for the genomics study. And you see in 20 years down the road that the genomic projects, the research in genomics, it's taken off and it flies very quickly. And even a year or two later, when this who is this guy again? I can't remember his name. Has come up with the ideal of personalized medicine. Who is this guy? I can't remember his name again suddenly. But anyway, so that is how it works. And then we see a lot of large initiative, large genotyping population projects actually are taken off, or to a stage that we look at 1,000 genomes, and then in Singapore look at 10,000 genomes, and even in in China, looking at 100,000 genomes of Han Chinese, and UK Biobank, and etc. So a lot of the large human genome sequence project it's taken off, and everyone jump into the human genetic and human genome sequencing, and almost everything, every living organisms under the sun is being published, is being sequenced. The question is that after sequence, what do we get? So we get sequence, and then obviously we get DNA sequences. But what does it mean underneath these DNA sequences? So that is the questions that people really do not understand. And many times I say that you understand DNA sequences, you run PCR, but that doesn't make you a geneticist, because at the end of the day, you have to understand what a role of a geneticist and what does it mean by a genetics subject. Yeah. So what exactly is genetics? To me, genetics, it's a study that look at the differences or the similarities of DNA sequence, obviously from the same species. In this case, we're talking about human and, well, Homo sapiens. And then we look into the questions of what makes you look different from me. Yeah. And, why Donald Trump doesn't look like orangutans, for example, I'm saying. I always make Donald Trump as a joke of mine. And then if you and I would look different, then what would be the consequences of these differences? So that is basically what genetics is all about. And as a geneticist, obviously, what we want essentially or eventually is to correlate our traits or phenotypes in a more specific term with our genetic information. In the other word, we are trying to figure out or sort out what does it mean of the DNA sequences or the structure of the DNA being aligned in such a manner. So that is our role. We look into diseases, for example, as a phenotype, and what genes, for example, would play a role in contributing to the disease development. For example, abnormal hormones levels, resistance towards COVID-19, and you name it. So that is the role of we looking into that. But in order to look into this ultimate goal as a geneticist, there are three different manners. One is to look into experiment based, in vitro or in vivo or knockout, you name it. Yeah, there's no direct applications to human because you can't grab or you can't hijack a human and put in the lab and do all sorts of experiment on his or her body. Definitely, you have to apply cell or animal model, and it often involves high experimental costs and is very time and labor consuming. Information that you got from experiment is entirely different. You are looking into pathway, for example, or what genes or what mutation that would be involved in the disease pathogenesis. There are two other different methods to understand the role of genomic in phenotypes. The second one is the most commonly used approach, that is genetic association studies. Yeah. That is the most common and easiest and most straightforward manner. All you need to do is to grab two groups of populations. One is a case where these individuals, this group of people would express the disease traits. Another group is control, where you don't have any disease per se, and then you mash it up and you run a statistical analysis most of the time, chi-square, yeah, or sometimes t-test, you name it, and you see whether that particular gene or mutation or haplotype, genotype, you name it, that would be significantly different between the case and controls. But the drawback of this particular approach is that you will need to have a large number of samples, and it relies on the observable traits, whether you have disease or you don't have disease, and because it involves a lot of strong statistical power. That's the reason why you need a lot of samples. And we talk about a lot of samples, we are not talking about 200, 300, we are talking about thousands of samples. We are talking about power of studies here. And obviously, there are a lot of other confounding factors. For example, that whether it involves different ancestries, different intermediate phenotypes, for example, I'm talking about hypertension in this case. If you know hypertension, they may have salt sensitive or non-salt sensitive. Yeah. So these are the confounding factors that may influence eventually the results of your statistical analysis in genetic association studies. And third method, essentially, is something that is very much relevant to the second approach, and that is from population genetics perspective, we call this evolutionary genetics, or in a more what we call as the hi-fi manner, we call this as the Darwinian medicine. Yeah, we look into a population, human population, as a nature, in the nature as an experiment. Yeah, because that's human as a model, and throughout the process of evolution, they have they have experienced the adaptations to different environments, and therefore shape their phenotypes. In this case, for example, resistance against malaria. I've always used malaria as a typical model, a classical model, an example to the medical students. Yeah, those with sickle cell anemia, for example, they are resistant against severe malaria. Yeah, because you have plasmodium cannot attach to your red blood cells and therefore being washed off. So that is a very classical models of natural selection. And that's classical model of how we understand the disease physiology. Yeah, but having the evolutionary genetic studies is not the only method, because it comes with disadvantage as well. Yeah, at the end of the day, in order for us to confirm the role of genomic in phenotypes, the three approaches have to be sort of reflecting to each other. It has to be answerable to each other's. Yeah. So I'm not saying that one is actually better than the other, but it all depends on your preference, expertise per se, and we cannot deny that they are all interacting with each other's. Yeah. The findings has to be interacting with each other's. You got a gene from natural selection, population genetics. You have to apply it in a case control cohort, for example. To confirm that, hello, can you hear me?
Yes, Prof.
I don't know what happened. I was being asked to force quit something. Sorry about that. Yeah. Anyway, can you still see my screen?
Yes, Prof.
All right. So I'm trying to say is that when you confirm a gene that may be associated with certain disease, then the next thing they probably want to do is to run it in vitro or in vivo to confirm disease pathogenesis. Yeah. So you see how this, I don't know what happened to that. So you see how these three approaches are interacting or interlinked with each other. Yeah. So do not ever say that one is actually better than the other. Now, understanding the role of genetics. understanding the role of understanding what does it mean by genetics and the role of a geneticist. That's where I come into the outline over here today, which I hope you understand better. How does the genetic structure Malay look like, and what is the implication for us when we look into the disease mapping efforts? Yeah. Now, when we look into population genetics, one thing that we always want to keep in our mind is that out of Africa, that is the theory of origins of modern humans. When I'm talking about this, is the modern humans. As a matter of fact, we know that even ancient or archaic hominins many times were originated from the center of Africa. So the moment when modern humans moved out of Africa to different continents, they started to have adapted to different environments. For example, in this case, colder climates, or all the way down to tropical forests where you have hot and humid climates, and then forests, and you have different diets and exposures, different even UV exposure, and to a certain extent, they may have met with the archaic hominins, and they have intermarriage or hybrid with each other. So it's already been proven that modern human carries at least 4% of the archaic human genome in our body. So there has been intercross of different hominin species during our modern human evolution. So all these actually shape our traits. How do we look like today? Yeah, and these do not take just 10 or 20 years or 100 and 200 years, but it takes more than 10, 20,000 years, or even up to 100,000 years. Yeah. So modern that is how the hypothesis of the outlook of Africa, and if we zoom in more focus into Southeast Asia region, that you actually see that Southeast Asian is a very interesting, highly heterogeneous, highly diverse region with highly diverse human diversity. Yeah, and they have been influenced by many different populations throughout the rest of the continents, and that makes us the admixed population in this case. And you really look into that, the very early work of my which I had the privilege of joining the Pan-Asia SNPs Consortium 12 years ago, that the Southeast Asian populations are essentially quite admixed. You see a different colorful, which represents different genetic structure that derive from different ancestries. Yeah, so you see they are actually quite highly admixed, as very different from other populations, like for example in Europeans or in Africa, where you actually see one or two colors. Yeah. So and if we zoom in further into the Malaysian population in this case, you see also a very highly heterogeneous population inhabiting a very rather small region in this Southeast Asia. Yeah, in Malaysia, from north to south to all the way to Singapore, Perlis to Singapore is only approximately 700 kilometers. Actually, very small region that we have here. But these small regions, at least in Peninsular Malaysia, give us a very highly different, phenotypically different native populations. Yeah, you have Negritos here, which are inhabited in the northern Peninsular Malaysia. You have Senoi people, which is at the central part of the Peninsular Malaysia, and the Proto-Malays, for example, at the southern part of Malaysia. And obviously, we have the native mainstream Malays. Yeah, Norita would definitely know who is he. Yeah, so you see a very different look of the native populations here, and if we run on the what we call as the analysis called the PCA, Principal Component Analysis, you see these populations are quite distinct from the rest of the East Asia populations or Southeast Asia populations. Yeah. So, and the work that I've done with my student, Shima, she was in IMB last, many years ago, that we actually see zoom in further in the Malay populations that represented by all these gray color bullets here. They are highly overlapped with the Cambodian population. This is something that is very interesting that we want to look into, and we are still looking into that. Yeah. But further dissection of the Malay populations, the modern Malay populations with the Cambodia, and this is what we got. We run an analysis called the Admixture, trying to dissect the different components from the Malays. And you can actually see, except for the Sri Lanka Malay here, yeah, I'll probably share with you 1 minute where does the Sri Lanka Malay comes from. Yeah, but if you look at the Malay population over here from Malaysia and Singapore and probably from Indonesia, and you will see that their structure, it's very much similar with the Cambodian population, except that the light color component, it's slightly different. The light color component actually derived from the islands of Southeast Asia. So that means that the ancestor of the Southeast Asia islands here actually contribute more to the Indonesian Malay over here, which is quite, makes sense in this case. But what I want to highlight here is the highly similar structure of ancestry structure between the modern Malays in Peninsular Malaysia and Singapore as compared to the Cambodian people. So we look into this and we dissect what happened. So how do we dissect? First thing, we try to find out whether the admixture is just by random or there was actually a timeline where the admixture happened. And just happens that in our calculations, we found that the date of admixture, the date where these different ancestral components meet up with each other, happens to be around 250 years to 1,250 years ago. Now, more recent analysis actually proposed that it was slightly longer time, approximately 1,500 years ago. Now, the different timeline is just because the different technologies were being used. The recent technology used a high density SNPs array. Ours one is actually less denser. I think around 500,000 of SNPs actually. But we managed to find to estimate the time between 250 to 1,250 years ago. So the timeline actually matched quite nicely to the history of the Malay land if you look into that. Yeah. So way back in 2,500 years ago, that's when the Malay civilizations of the Malay land actually occurred. And then you see different empires in the different timelines actually occurred and conquered the Southeast Asia to the Malay land, Malay archipelago. But one interesting part is that where the Champa populations, located in Cambodian and Cambodian area, Cambodian country region, were the one that actually been long lasting and conquered the Southeast Asia for a very long time, and all the way when we have the Malay people conquered our regions, and that also contributed to certain extent of the admixture or genetic structure of the Malays here in Peninsular Malaysia. So I cannot conclude that the Champa Empire is the one that contributing to the highly similar genetic structure between Cambodian and Malays as of now. But we definitely want to look into that. We are in fact in the midst of looking into the reasons why there is a high similarities between the two populations now. This is a work that actually mainly done on the Sabahan natives actually. But what I want to highlight here is on top of the very distinct structure of the Sabahan population, Sabahan natives over here, the Malays are also quite different. If you look into that, they're actually very different from Sabahan people, although we are, so to speak, Here you go, Malay Kelantan and Malay Singapore. Although we, so to speak, under the same roof of the Satu Malaysia over here, biologically we look actually quite different. Even the Malay Minang, which we collected at the Negeri Sembilan area, there's actually a population called Melayu Minang over here. Their populations are quite different, and we look into the history, they have around 2 to 3 generations being migrated from Palembang to Peninsular Malaysia, and then inhabiting or staying in the areas of the Negeri Sembilan. So a very short history in Peninsular Malaysia. That's the reason why they actually stay quite further apart from the Malays of Singapore and Malays of Kelantan. Yeah, but having said so, we look into that, we see that the Malays over here, which is in green color, actually were quite distinctly separated from the natives of Peninsula, native of Sabahan. In fact, native Sabahan actually stays closer to the East Asian people, which is the Han Chinese in this case. Yeah, so that actually indicates that the Malays, modern Malays, and the native of Sabahan will have a different history of migration to different place. Yeah, to a different place. Now, the message I want to deliver here, first thing is that there are enriched and mixed genetic diversity in Malaysia. Yeah, and then they have distinct genetic patterns being observed. And the third one is that, which is quite obvious in the analysis that we published earlier on, the mainstream Malay would have at least 4 genetic ancestry. At least 4 genetic ancestry that forms the modern Malays nowadays in Peninsular Malaysia and Singapore, perhaps some part in Indonesia as well. Now, I want to share with you just now, I forgot to tell you that where does the Sri Lankan Malay comes from? This Sri Lankan Malay essentially were from Indonesia. Yeah, they were actually been sent to Sri Lankan by the Netherlands troops. Yeah, they were sent there for a war in Sri Lanka and about 200 to 300 years ago. Yeah, so they still keep the Malay culture. They still speak Malay language until now, but they are, because of separations of few hundred years and the admixture, and they have intermarriage. You see nowadays that the Sri Lankan Malay will have more in South Asia ancestry components as compared to other modern Malays nowadays. You can easily Google the Sri Lankan Malays, and you can actually take a look at their history. It's a very interesting history of the Sri Lankan Malay. Let's look at how the genetic structure of the Malay population as a whole. So we further go into that and dissect how does the structure look like. So we collected the populations, all the modern Malay population throughout the Peninsular Malaysia. So we actually run a population differentiations, and we divided them into three different regions: northern part, central part, or the southern part. We found that they have a very close genetic similarities. Their population substructure is actually very minimal, which is fine. But having said so, when we look into further details on principal component analysis, we actually see a bit of clusters between the north and south. Yeah, the Peninsular Malaysia and Singapore Malays actually quite separated distinctly. Yeah, and the Peninsular Malaysia represented mainly by the northern part of the Malays. Yeah, and we actually see that the northern part of the Malay had a closer genetic affinity to the South Asian or to the Indian people. Yeah, and that actually give us a bit of the puzzle of what's actually happening here. We have not found out. But a further dissect, and we run on again the analysis called admixture. Yeah, you actually see that there's a very significant difference in the blue and red components over here. The green component is represented as the central Peninsular Malaysia component. But if we do remove this component, we actually see that it's actually north and south. There's actually a very obvious incline of the red components. Yeah, and that actually represents something here where the genetic components were correlated with the geographical coordinates. Yeah, so there's actually an incline between the northern part and southern part of Malays. This is what we try to say. Although the Malays look alike, very closely similar, but if you look at into details, their biological structure, they are distinct. And how much that these distincts actually cause? Yeah, this is just to show you that the correlation between the geographical latitude and their principal component actually shows a very significant correlation. Now, how much different is that going to be? We try to look into this simulation. Wow, where is the simulation part? Yeah, okay. So we how much we look into that, that's the simulation. And that is the second message that I would like to deliver over here before I move into the third part. Yeah, the second message here is that population structure has occurred between the northern part and southern part of Malays, despite the different region. And there's a possible attributions to either the genetic history or maybe to the geographical latitude. Yeah. Now, these differences that we look into that before, I would like to explain how much impact that we create. Let me just spend 1 minute and explain to you what does it mean by the impact over here. Now, assuming that you have one population ancestry which carries a different prevalence of allele. Yeah, allele A, for example, with 60% and the mutants with 40%. Now, this population of ancestry move on to create two different subpopulations, population 1 and 2, which we call as the migration bottleneck over here. And this migration does not happen in random at that time. Yeah, most of the time. So it could just happen that population 1 would carry 80% of the mutants instead of the wild type, whereas population 2 would carry 90% of the wild type instead of the mutant. So a very obvious differentiation between the two populations here in terms of their frequency. It happens by random again. Well, it does not happen by random. It happens by, we have one population move up to another. It's like a migration happens. Yeah. So this thing would just happen. What we call in genetic is the genetic drift here. Now, if we were to take these two populations and we run it in case and control study, genetic association studies, you will see a significant bias between the case and controls, where you have case is likely to have 76% of wild type, when controls you have only 34% of the wild type. So if you were to run a case control study, chi-square analysis, you are creating a bias over here, because there's it's an uneven frequencies of your wild type and your mutants. Yeah. So if that actually happens, what that will definitely influence your statistical outcome. And that's the reason why we often want to have as homogeneous as possible for our case and controls. Yeah. Now, because of this, we ran a simulation between the southern part and northern part of the Malays. Yeah. What simulation that we did? We take approximately 40,000 SNPs and then we ran a case control study. Just a case control, assuming that the southern part of the Malays southern Malays are controlled. So we run on that and then we estimate the potential levels of false positive that may occur. In this case, represented by lambda. We call this lambda as an inflation indicator, yeah, inflation factor. Yeah, inflation factor. So any of the inflation factor that is less than 1.1 is considered to be acceptable, but anything that above 1.1, that is going to be a trouble for us. Yeah. So in this case, when you have small population size, that is acceptable. But when you have small population size, your power of study is actually low, much lower than you are supposed to have. When you increase your sample size, you tend to get high chance of false positive. Yeah, you tend to get high chance of false positive, and this is only what I'm talking about, 40,000 SNPs. So if you increase your number of SNPs, you increase your case control study, your lambda value will definitely be affected in this case. And that is what I mean by having a statistical impact when you have not controlled your population ancestry very well. Yeah. In this case, we just have to be a little cautious when we run a case control study for the Malay populations. This is the message I would like to deliver over here. Now, in order for us to minimize that, there's one method, that is to identify a markers called the ancestral informative markers, AIM. Yeah. So how does it work? It works by identifying the genetic markers that is able to differentiate between the southern part and northern part of Peninsular Malaysia, for example, in this case. So one way to identify that, then you know that how much substructure that would that you have in your case control population, or you can even remove these, so to speak, as AIM markers, and then if you rerun your case control study and see whether it affects your p-value or not. Yeah, but the unfortunate thing is that the AIM panels for Malays are still not available at the moment. Yeah, there's an AIMS panel for modern Malays as compared to the Southeast Asian populations, but there's no AIMS panel for us to differentiate between northern part and southern part of Malays. Yeah. Why is that not? Because in order to create this panel, you need a lot of samples. You need full genome sequencing of the northern part and the southern part of Malays before you are able to identify an effective panel of the AIM. Yeah. So in Malaysia, you don't have money to do that because genetics is always considered to be an orphan disciplines in most of the grant applications. Yeah. And it's pretty unfortunate. So as of now, we don't have the AIMS panel yet. Now, having said so, not to say that the mixed population like Malay will have difficulties or running a case control study. No. As a matter of fact, there's a method of identifying a gene using case control study from the Admixture population. In this case, we call as the Admixture gene mapping method. Yeah. So we use the African population as one of the example because African American has got the admixture between African and American and European. Yeah. So if you are able to identify the segments that derive from African or which segments that derive from Europeans, then the signals that you identify from your disease association study that gives you back to the ancestry, which ancestry that it came from, that will allow you to identify your gene mapping or your susceptibility of the mutation to a disease much easier manner. But obviously it involves a lot of advanced statistical analysis. Yeah. Now, before I deliver the third message over here, just like to share with you the case control study, the genetic association signals that you identify. I emphasize it, it's a signal. It is not the genetic mutation that causes you the disease. It is only the one that is associated with the disease because the inheritance recombinance comes along with that particular genetic variation with the phenotype. Yeah. So to identify a mutation that causes a disease, you need a lot more efforts in order to do so, which include, like I say, in vitro and in vivo studies in the lab. Now, these potential association signals are essentially identified when we call this as a signal of natural selection. It's another way of the signal of natural selection, but in a much, much weaker manner. Yeah. So that's the reason why I say that genetic associations and population genetic studies are closely related to each other. And that comes to my third message over here. The population substructure may lead to spurious association signals, yeah, that may affect your p-values of your case control study. And developing the AIMS panel would be helpful. But having said so, Admixture mapping is an option, and therefore Malay population, if we utilize the genetic information well, we will still be able to identify or use this population for gene mapping studies. And that is relatively important for our approach or for our aim towards precision medicine. Yeah. So, and just would like to share with you a work that is being done by a student of mine, Hasna. She used to stay in IMB as well. I suspect many of you would know her. You see that these two gene is a very common genes that involve in the system called renin-angiotensin angiotensin system, which controls the salt and water retention, homeostasis in the body. So you'll see that the common mutations that we identified here are strongly correlated with the latitude if you look into the global populations. Apologies, the dots are actually way too small for you to identify. Yeah, but what I want to share with you here is that the Malay population from Peninsular Malaysia, which also include the Negrito population, orang asli, their SNPs frequency here, their mutation frequency are highly similar or make no difference from those that are staying at the tropical region or the equators, which include the African populations. Why? Because these genes has an experience of natural selections out of Africa. They have adapted to different temperature and different climates and exposure of the UV. Because the way you move all the way up to a lower or colder climate, you would have less efficacy. You will want to adapt to an environment where you have less sweating and therefore you have less sweat loss. Whereas those that are at the extreme hot climate over here, you need to have an extreme efficacy in your water and salt retention. Otherwise, you get your water loss, your salt loss, and that may cause to fatals. Yeah. So those genes that are in favor of this hot environment or mutation that are in this hot environment are always being preserved. That is the reason we see a strong correlation of the mutation with the latitude coordination. Yeah, so that part of being shown here is actually a typical example how population genetics and disease physiology can be all linked up together, right? So it's a very different study over here. I don't know whether I still have time. Ita, do I still have time for it? I can skip this part if there's no time.
I think your talk until 1:30, the Prof.
Okay, so in this case, I will still have another maybe 10 minutes time to share with you what natural selection is all about. I've been talking about population genetics, evolution, and natural selection. For my 30 odds of the slides here. But natural selection is actually a primary force of evolution. Why do I look darker than you, for example? That's because I'm being exposed under the sunlight a longer period than you. When I say longer period, means longer many years ago. Yeah, because why? Because my skin pigmentation has evolved in the sense it's changed and adapted to the exposure of UV under the sunlight, and therefore it becomes darker. This is one example. And don't be surprised that we are still evolving. For example, there's a vein changes in terms of anatomy, yeah, of our hand, and also the wisdom tooth is another classical example that people has been talking nowadays. Some people will have wisdom tooth, some people will not have wisdom tooth, and you see the younger generation, yeah, the chances of them getting wisdom tooth is actually much lesser. The frequency, if you compare different generations, that is why that is a sign of the evolution. That is a sign of natural selection, because nowadays people don't have wisdom anymore. So that's the reason why we don't have wisdom tooth. Yeah. Look at our politician, how much wisdom that we have, basically. So that is a part of natural selection and being proposed by Darwin. And don't be surprised, yes, we are still evolving. Yeah, and we look into that. I'm just going to show you how natural selection works. I've not done this in Malay. Well, I tried to do this in Malay, but then unfortunately, the admixture signal is too strong that has prohibited me from identifying a strong natural selection signal among the Malays, and then therefore I not been able to identify which traits are they being involved. But the North Bornean native, the Burmese from Sabah, have a very unique structure as I see here. And then when we run on the natural selection analysis, there's a region shown here in the chromosome 6, chromosome 6 MHC region, a very interesting, very strong. So when we see it's actually peak over here, that is a signal of the selection here. Each of these dots represent one SNP probe. One SNP probe. And we see actually a shoot here, a volcano sort of things here. It shows that in this whole region, a very long haplotypes over here, these signals are being selected, which again says that there's a recent natural selections happen. So when we found this signal, we thought that this is a very interesting thing, and we wanted to publish. In fact, we sent to Nature.com. So the editor actually rejected because they said that MSC region is too common, and therefore they are not interested in looking to that, which is a disappointment. So we sent to another journal, to MBE, Molecular Biology Evolution. I think the impact factor is about 16 by now. We went through in a review process, and then the same thing commented by the reviewers. We went through the editor, and the reviewer commented, but unfortunately it's the same comment because it's MHC region. And that's why the geneticists do not look into that anymore. But anyway, to me, I think this region is very important, although it's a very common region of undergoing the natural selection. Why am I saying it is important? Because when we identify this particular signal here, now this is just to show you that how long is the signal that we found here. It's almost 2 megabases. And then when we zoom into 2 megabases, we zoom in approximately 13 kilobase pairs, and of which, based on this particular signal, this particular state, of which the 13,000 kilobase pairs of the haplotype that we identified, they are very homogeneous. Imagine that 13,000 kilobase pairs, actually very long sequence. So how much chances that you get? How many combinations that you would anticipate to get based on this 13,000 kilobase pairs? Almost every nucleotide has got a chance of being a variant. So we would definitely get a different kinds of combinations. But in this case, very interestingly, we only able to identify 7 haplotypes. There are 7 patterns that is only found in this 13 kilobase pair length of the DNA sequence. So, and these 7 haplotypes, we call this, majority of them lies in group A, haplotype 1, 2, and 3. The rest, 4, 5, 6, 7, have very low frequencies. So we know that this 1, 2, 3, especially haplotype 1, is the most common DNA sequence in the Sabahan people. And we run an analysis and compare to the rest of the population in the world for these 13,000 kilobase pairs, and it happens that only unique to Sabah natives. So with that, we estimated the time, and we actually see that the time when this signal shows up, when this signal shows up is about 5,000 years ago. And that is when the agriculture technology been expanded in Southeast Asia. And why is this important? Because that is when the humans, the modern humans, started to have heavily intact with the Plasmodium species. That's when Malaysia, the modern humans were being infected heavily by Plasmodium species, in this case Falciparum. And I think that the signal shows up differently among the Sabahan people, not because of Falciparum, but rather because of the knowlesi. Because Plasmodium knowlesi has occurred in Borneo for more than 20,000 years. If you look at the papers published by Professor Babu Singh. So therefore, I think the signal is actually related to the Plasmodium knowlesi, and we are still running genetic association studies in collaboration with my Sabah UMS friend. looking to verify or to validate the signals that we found. Yeah. So with that, I would like to just put the end of my presentation here by giving you a few messages here. I saw Prof. Chen is around here. I'm sure there are many other colleagues over here. Yeah, I hope this message can be delivered across to many colleagues here instead of students. But students, please keep in mind, first thing, it is very important to understand classical genetics. Yeah, because by understanding classical genetics, that is how you're going to utilize that into your disease physiology and this gene mapping. However, the emphasis nowadays is insufficient. If you look at what MQA does, yeah, it says that genetics can be everywhere and it is no longer a subject, which is a set. Yeah, there's a lot of underneath messages to be delivered instead of we understand what Mendel tells us. Second thing, the fundamentals of classical genetics should be emphasized, and we have not emphasized that. Yeah, we have not emphasized that. Many times I blame those who are not geneticists and they declare themselves as geneticists because they've got funding and then they have run the research and then that's the end. And that's not the end at the end of the day, but because of they don't understand what genetics is all about, and that becomes the end, which gives a bad impression to the stakeholder, which I don't blame the stakeholders anyway. But I wish to emphasize here is that the fundamentals of fundamental research, especially for classical genetics, should be emphasized, and that's what we are doing out there from Malaysia. Look at Europe, look at the States, yeah, look at Chinese, yeah. So funders, you should redefine what is meant by fundamental research, or what does it mean by translational research and applied research. And even now, FRGS, we are talking about pattern search, we are talking about how your research can be utilized, and there's no longer fundamental, well, there is still, but very minimal fundamental components that are being emphasized now for our fundamental research grant scheme, which is a set again to me. Yeah, look at Chinese. They have 100,000 genome sequencing done on the Han Chinese people. And you know what? 90% of the funding comes from the pharmaceutical industries. That is how they emphasize their fundamental research in order to catch up to the West people. So that's the end of my slides. I'd just like to thank a few people over here, which include the FRGS and the MOSTI grant, which I've got. Yeah, the FRGS grant and MOSTI grant that I've got, including the grants I had last time when I was in IMMB. Yeah, and also my Chinese counterpart from Chinese Academy of Sciences, who had appointed me as the visiting professor for a year. And funded me for the many of the studies which I mentioned over here. My university, UCSI, and also definitely not to forget UITM, Sungai Buloh campus, and the fellow friends from IMB. Many of my earlier works were done from IMB. Yeah, and obviously not to mention, not to forget my mentor, the late Dato Professor. The late Professor Dato Khalid Yusuf, who has just passed away several months ago, and other friends from NUS, from UMS, and to your Deputy Dean, Associate Professor Dr. Tuhaira. We have been in communication, and she was with me in the sampling trips for the last 10 years actually. Yeah. So and without these fellow friends, I would have done a great job in the population genetics. With that, I would like to just thank you all for your attention.
Okay,
Rita,
thank you, Prof. Yeah. Yes, thank you, Prof. Yes, Prof, I still remember picture, the man that you show in the slide, Mak Rose from USM, right?
Yes, you're right.
And I hope I can see him again.
Yeah, I don't know how you see now. He was not well for a while, and I think he's okay now.
Okay. Okay, now we open to any question from the floor. You can straight away ask the question to Prof. Ho.
question? There are only two reasons when there's no question. It's either they fully understood or they don't understand at all.
Someone told me that your talk is very advanced, Prof.
Oh, yes. It's not that the talk is advanced, yeah, but rather it is because that the students, you guys, are not being exposed to what is supposed to be exposed. In this case, the classical genetics, the Mendel's genetics that you are supposed to be exposed, but you are not being taught in your study time, your students' time, which is something that I would like to sort of again re-emphasize.
Good afternoon, everyone, to Prof. Thank you very much for your impressive and excellent presentation, and thank you for accepting our invitation to speak at IMB platform. Prof, I have one question. You know that Malaysia is also a mega biodiversity country, that we have a lot of flora and fauna. And do you think that we should have our own genetic information database to document our local flora and fauna for the genetic studies as well? I'd like to hear from your opinion. Thank you, Prof.
Thank you, Dr. Hill. Very nice to see you back again. Yes, indeed. Again, like I said from the beginning of my talk, it's actually a pleasure of mine coming back to IMB here, although in virtual, and to see all my fellow friends, here. And back to your questions just now. Indeed, yes. And if I'm not mistaken, the Academy of Science Malaysia actually has been starting an initiative of what we call as the BioD project database. It just happened that I'm actually sitting as one of the working group member for the Malaysian Open Science Platform. So one of the sub-project, there are actually two sub-projects. One of the sub-project is COVID-19 database. The other sub-project that they have actually been planning but not being moved because money elect basically, and that is a BioD project. And I fully concur with you, your idea of having a preservations of the data for the Megadi in Malaysia, because that's the precious and that's the asset of Malaysia.
Yes.
Yeah. Because if you want to work with outside there without having a bullet, that's it. And the bullet that you have here in Malaysia is a Megadi that you have.
Yeah.
I hope I answer your questions. I agree with you. But yes, there's something that's trying to work on. But again, Malaysia always talk about money, and Malaysia has never said they have money.
Yeah, that's interesting, Prof. Yeah, we really hope that we will have a center that can store the DNA of the people as well as the local flora and fauna. As you said, I totally agree with you that we need the bullet. Yeah.
IMB can start that.
I really hope, but really need to know the mechanism, the pathway. Prof, really need to need your help. Yeah.
I'd be very happy to speak to you all in a separate platform pertaining to this planning. But definitely, yes, I think IMB would have the capability of starting something about that, actually.
Okay, thanks, Prof. Thanks for believing in us. Thank you. Yeah.
Thank you.
Okay, Prof. I think there's no more question.
Sure.
Okay, so I end today's research talk with thank you to everyone, especially Prof. Boon Peng, for the time he took to share his research experience in the population genetic of Malays and its medical implications. So I hope you can join us again, Prof.
Yeah, sure. Well, definitely. I hope so. But let me apologize to you all for the hiccups in the beginning of the talk, because I've just not get used to the Google Meet thing. Anyway, I apologize for that. But yeah, I will spend some time again, hopefully, to make you guys a visit in IMB soon, maybe after Chinese New Year.
Okay, no problem, Prof. Okay. Once again, on behalf of the IMB, thank you, everyone. So goodbye, everyone. Stay safe and see you in the next time.
Thank you. Bye.
Thank you, Prof. Thank you, Norita.
Bye.
Thank you, everyone. Thank you. Bye-bye.